Thank you everyone for joining us today, and I'm so excited to hear such great presentations from today morning. And now we are in our live Q&A. I would like to welcome all our guest speakers today in this live Q&A session. My first question is to Sydney. Sydney, this was a great presentation, and first time I heard this word productization. And in this step of productization, which particular step you feel is more challenging, or where you see we need to spend a lot of time, whether it is about regulation, about materials? So which step is most critical or most challenging in productization?
Hi, thank you. Thanks, Vibhu. And I want to thank the organizer for having me on this webinar today. So on this question, if you remember the presentation, we always start with the assay optimization. The key here is really when we talk about end-to-end productization, we really just start with the use case, and then that should drive what sort of optimization we need to do as early as possible. And therefore, we have this thing called agile iterative. In other words, everything we do, we always need to check back with the use cases. So the earlier we are able to optimize the assay for the purpose of the use case, and the earlier we are able to get some sense of whether it can fulfill the performance required for that specific use case. If it can't, then we need to be able to pivot and to change and see whether we are able to then adapt that to a different use case from product development perspective. So I would say the important thing is always to make sure that the optimization is done properly and as early as possible. And what that means is really to see, first and foremost, whether your assay itself is reproducible functionally. So it needs to be, you have to understand that we're talking about an IVD. So it needs to be a test that is robust, that is something that any lab, any operator working in a lab is able to take your instructions for use and just follow that through. So the robustness, the functional reproducibility, doesn't really matter who runs it, it will give you the same performance. So those are the things that we focus a lot on when we're talking about optimization of an assay. So if you remember the anatomy of a diagnostics test, assay is only one of the components. That's also where you need to work into the use cases, and therefore you know what are the other components you need to add in. So at the end of the day, it comes down to the technical performance. If I can sum up, it's really make sure that you are able to get functional reproducibility as early as possible, and make sure that the assay itself is robust enough before you take the trouble to build the whole diagnostics test around it. Thank you.
Thanks, Sydney. Sydney, there is a follow-up question, and if I go back to yesterday's presentation, so Dr. Jerome came, so he shared with us that the whole vaccine process, it takes many years, but new expedited process can get it done in say 12 to 16 or 12 to 18 months. So from a diagnostic perspective, what do you see? Like, are the tests getting expedited? How the regulatory bodies are addressing this challenge that we have these innovative tests, but how to bring those tests faster to the public?
Yeah, so I think from a regulatory standpoint, as we all know that there are all these emergency use authorization. So in Singapore, HSA has something similar called Provisional authorization. So these are actually authorizations that are given for IVDs they are not equivalent to a full registration because they are just there are certain specifications from regulatory standpoint that just need to take the time, and in an emergency setting, those are specific technical specifications that the regulators are willing to say that at the point of registration, I can't give you full registration, but it's provisional. What that means is that as you are rolling out, you need to keep checking back with the regulator on a regular basis and keep updating the information. So we are, I would say, for diagnostics and I suppose maybe for vaccine and therapeutics as well, in the case of such an emergency situation, that most regulators are able to deal with different provisional and emergency type of treatment of the different samples without different tests, without compromising on the quality. The very, very basic quality management system, the very, very basic how you do your analytical verification and clinical validation, those are basic information that even in the emergency settings still would need to be furnished. But time generally can be accelerated because of the situation where the regulators are willing to say, roll out and then keep checking back with us on some of the other details like stability data, for example, which could take longer than, the months that we need. Yeah, thank you.
Thanks, Dr. Sydney. I will go to Professor Rajiv Khanna. Dr. Khanna, one question which is coming from the audience here is, in your research, VCCMV, how do you see coronavirus and influenza impact on transplantation, especially as the COVID-19 infections are increasing so much? Is transplantation getting impacted by these COVID-19 infections, and how serious it is? Professor Khanna, can you hear us? Okay, maybe we are not able to connect with him. So I will go to Professor Mashimo. Mashimo-san, CRISPR-Cas3 and CRISPR-Cas9, we are working from Merck side on these areas for a long time, and CRISPR-Cas3 look quite exciting for both gene editing and diagnosis. From the sensitivity perspective, what difference you see in Cas3 based detection versus Cas12 or Cas13?
Yeah, thank you very much for your question. It's DNA editing. Cas3 is DNA editing enzyme, so it's like Cas12. So the efficiency or sensitivity is equivalent to the Cas12.
Okay. And yes. So this is a question, but if we bring this test, are these tests good enough for diagnosis, or do you suggest that it will be good for surveillance or epidemiological studies?
Oh yes, that's a good question. So we are now actually making a CRISPR-Cas3 protein in the faculty. So we have to produce a great amount of the protein. So we're still waiting for that. But we checked some samples from the hospital, and actually it's very as you know, the CRISPR-based diagnostic it's very depend on the PCR. So I mean RT-PCR protocols. So it's also if we are positive for RT-PCR test, then we have the same result by the CRISPR-Cas3 diagnostics.
Thanks, Mashimo-san. Dave, if you can hear me, you are also working on a CRISPR-Cas based detection kit. What are the challenges you see in getting it to the commercialization?
Oh, that's also a good question. We have to rush because we haven't yet permission from Japanese government, PMDA, like Japanese FDA. We are now connecting with them, and we are also collaborating with diagnostic companies which are manufacturing our enzymes. So we still need a few months, but maybe in the winter season we will have again the pandemic situation. So we will do it. Thank you.
Okay, Mashimo-san, one question. It's more of I will not say technical, but you moved your lab from Osaka to University of Tokyo, and it was during pandemic. It must be quite challenging for you. So how you manage this entire thing, getting research started again from one university to another in such a challenging situation?
Thank you very much for passing my personal things. So yeah, it's very challenging, and in this case, luckily we have the two labs in Osaka University and Tokyo University. So we gradually moved from Osaka University to Tokyo University. So we still have the lab in Osaka. So in my case, it's okay. Thank you.
Okay. No, I like both Osaka and Tokyo. So next time I am there, I'm gonna see you.
Thank you.
Dr. Khanna, can you hear me?
Can you hear me? Hello?
Yes, I can hear you.
Okay, that's good.
Yes, I can. Okay. So, Dr. Khanna, I'm just repeating the same question again, that we know infections can be the major reason for complications for transplantation. And with the COVID-19 infection increasing so much across the globe, how do you see its impact on all transplantation-related work?
Yeah, it's been a major challenge particularly for stem cell transplant, where a lot of donors who donate stem cells are international. For example, a lot of donors recipients, patients who are actually expecting the stem cells, they often come from Europe or Germany or some other country, and those shipments have literally been completely cut off, and there's hardly any, and these stem cells are coming across. And same thing is happening with the organ transplant as well. There's a lot of challenge with this COVID-19 causing a lot of delays in transplantation. There has been push towards moving away from allogeneic stem cell transplant to more what we call haploidentical, where the sibling or one of the parents actually becomes a donor, where you've got a 50% match of the HLA. So that's what most of the centers are trying to develop protocols on that, and some centers have already switched to that. Other centers are looking at that. There are additional complications of that because haploidentical being partially matched, it can raise a lot of other clinical issues. For example, if you don't deplete T cells and the B cells, you can get graft versus host disease. But if you deplete the T cells, which is absolutely essential in haploidentical transplant, you get viral complications, as I've shown you a few of the examples in pediatric cases. I've shown you some of those ones.
Okay. Another question, Dr. Khanna, for you, I see is T-cell therapy. It's something which started in last few years, and people might not be well aware about regulatory requirements, etc. So can you elaborate around the regulatory requirement for T-cell therapy?
Yeah, sure. I think the best example for that is the more recently approved the Novartis product, which is the Kimra by FDA. It is it's a hugely challenging process because to manufacture any living drug, so it's like these immune cells are living drugs, and to deliver those from the patient or from the off the shelf, requires a lot of regulatory compliances, and we have a dedicated facility. You have to have a world-class cell therapy manufacturing suites available to you. Your regulatory in terms of quality assurance, quality control systems have to be in place. And of course, the most important part is to be able to the supply chain, which is absolutely crucial. You do not want your raw material to come to you at a wrong time, and then your material, when it's manufactured as a drug product, to reach to the patient at the right time and the right place. So all the supply chain needs to be very carefully managed, and that has taken us, well over a decade to streamline those things. So we have patients actually not just within Australia, but also internationally. We treat patients in Hong Kong for certain clinical trials, and it's a very complicated process. But in Australia, the Therapeutic Goods Administration, which is similar to FDA, has given us a manufacturing license for clinical trials. And similarly, we also collaborate with a biotech company in US, Atara Biotherapeutics. They have built their own facility. A lot of companies are now building their own manufacturing capacity to deliver these cell therapies for patients. So it requires a lot of work.
I can understand that, and people probably want more work from you, and that's the reason I see a question that Professor Khanna, will we be having some COVID-19 related kits from your side? It's a very specific question.
No, it's a good question. In fact, we already been working on that. One of my colleagues here, Dr. Corey Smith, he's actually developed a whole blood test for COVID, similar to QuantiFERON CMV, and we can actually we are running that test quite routinely now, but not as a commercial product, but more as a research reagent, and you can rapidly detect within 24 to 36 hours the immune competence of the patient can be determined very quickly by using similar QuantiFERON type of assay. Yes, we are developing a kit for COVID as well.
Thank you. And today I first time came to know that we are having Sherlock as a kit coming from one lab, and Detector as second lab, and Paluda from India, from Dev's lab. Dev, can you hear us?
Yeah, I can hear you. Can you hear me?
Yeah, so I'm quite excited, Dev, to hear so many detectives coming into the picture. But there is one scientific question which a couple of people are asking from you is, how sensitive is Paluda in comparison to other tests available, whether it is serological or RT-PCR based test? How sensitive is your test?
Right. So the sensitivity and specificity currently are being matched with the gold standard test, which is the quantitative real-time PCR test. We have a sensitivity and specificity of above 95% on Paluda, and this is not just what we tested here over almost 2,000 samples here in IGIB, because we're doing a lot of testing and we're improving. But now I think Paluda has also undergone third-party validation by the regulators, and I believe it's around more than 95%. In some cases, we get 100% sensitivity and close to 98% specificity. In sometimes it's around 96%, 98% on 2 different days or something like that. But our goal was to bring it as high as possible. In the process, we also learned a lot of different things, including the relook at what we call as a gold standard in the field, because several qPCR samples which are classified as a positive or negative gave different outcomes as well. We learned all these things during the way. It was a long drawn process. But I think in the end, we have a similar kind of sensitivity and specificity as the gold standard PCRs that are currently being used.
Okay. Dev, there is a related question, and I think you might be able to answer that. Can we differentiate between coronavirus infection and other influenza viral infection, whether it is from a symptoms perspective or a detection perspective? Yes, please share.
So this has been done as well. So it is 100% efficient in distinguishing between corona and other infections, which are of different similar symptoms, viruses. But what I would like to point here is that the principle of detection uses an enzyme which is called Francisella novicida Cas9, and it's engineered versions which we made. This particular Cas9 has the property of distinguishing between substrates which are differentiated by one mismatch. So the specificity is really, really high. So unless you have another sequence where there would be, around almost 90 to 95% homology with the target, which we are anyway taking care not to include in the design step. But even if there is an amplification of that kind, that would be differentiated by the specificity of the enzyme itself. And added to it is that in the Paluda step, we have at least 3 layers of specificity added. One is the reverse transcription reaction, which occurs with a gene-specific reverse primer for the SARS-CoV-2 RNA transcriptome, and then that produces an a cDNA, which is specific to the virus. We add another level of specificity with a positive or a forward primer, which is again specific to that same reverse primer, and then finally we have the specificity due to the Cas system itself.
Thanks, Dave. I will go back to Sydney. Sydney, what are the various products DXD Hub is able to launch around COVID-19 in recent past, if you can share?
So I think, so this is about the products that DXD Hub had launched relating to COVID-19.
Yes.
Okay. Yeah, so we have, so since the beginning of February, we have launched quite a number of products that are directly related to COVID-19. We started with the first one, the first week of February. We launched 42, it's a conventional RT-PCR product. I mean, now, of course, there are a lot of such conventional RT-PCR products on the market. But if you think back towards the end of January and the first week of February, there were very, very few such regulatory approved conventional RT-PCR tests on the market, and very, very few countries actually are able to secure such test kits for that diagnostics purposes. So it was one of the, so I would think that we were one of the earlier ones that were able to launch a regulatory approved product, which now has been deployed in, I think, over 45 countries, have regulatory approval in quite a number of countries as well. So that followed by CPAS. So CPAS is, so maybe let me just add on, 42 was a collaboration between A*STAR, the Agency for Science, Technology and Research in Singapore, as well as one of the local hospitals, Tan Tock Seng Hospital. We then followed that with another collaboration, which ended up with the launching of ELISA tests for specifically to detect neutralizing antibodies. So this was actually a collaboration with Duke NUS and the company GenScript. The product is called CPAS. So it is still the only product on the market that detects neutralizing antibodies. Of course, then we collaborated with a number of other research institutes, including our National University Hospital and University, to launch a point-of-care rapid diagnostics. It's a finger prick test for antibodies. So most recently, which is the one that I actually highlighted in my talk, was the direct PCR test. That was a collaboration with Defense Science Organization in Singapore. It's called Resolute. So that's a direct PCR test that completely is able to do away with extraction. So basically, it goes directly from the patient specimen into the PCR master mix and PCR. So it cuts down on, in fact, not so much the TAT, but really a lot of the workflow and makes it a lot easier to deploy. Now all you need really is a thermocycler in order to deploy a test like a direct PCR, like Resolute. So these are the number of tests that we had launched and deployed with our partners since end January, early February. And these are specifically for diagnostics purposes. We have also collaborated with a number of other private and public sector partners to deploy more reagents, so supporting reagents for COVID-19 and supporting tools for management and monitoring of patients in the ward. So these are the different projects that we are still working on quite a number of pipeline projects that are looking at different use cases. So I think for diagnostics, it's important to look at the use cases of the population, whether you're looking at symptomatic or asymptomatic patients. That would actually drive the performance that's required for your assay and how your test should look like in terms of the product form factor, whether it is done in the lab or whether you want the product to be able to done in the field. Yeah, thank you. Vibhu?
Yes.
Yeah, sorry. I thought I lost you. Yeah.
No, we can hear you.
Yeah. So basically, that's sort of sums up the some of the products that we have launched and also the pipeline, some projects in the pipeline that we're working on.
Yeah. Thanks, Ajay. One thing which inspired me a lot is, and Dave, you used a good word for that, COVID warriors. When most of the labs in academia were shut down, all of you guys were working in your labs to bring innovative solutions. So I just want to ask each one of you, what were the challenges you were facing? And I will start with Dave. Dave, you mentioned that some of the students, researchers, got even COVID positive during this journey of innovation. So how challenging it was and how you were keeping your lab motivated enough to work during this phase, Dave?
Yeah, that's actually a very important question. We sometimes put that right at the back burner because the pressure and the motivation to get something out that will be helpful sometimes dominates the mental health, the physical health, and so on. So, like I said, that yes, we had situations where the lab members were themselves getting infected. We were going and collecting samples from areas which were not a very safe place to go. But I wouldn't call them as challenges because we're really looking forward to finding out a solution that could be low cost and which could be effective because the assay doesn't require the need for a quantitative real-time PCR machine. But I think the challenge that we really faced was actually to get over the regulatory clearances. And sometimes the regulatory clearances for new technologies benchmarked against existing or old technologies makes it really complicated to put a threshold as to what do you qualify as a successful approved technology. And we learned it the hard way. We realized that we were benchmarked against quantitative real-time PCR, which itself till we did almost in IGIV now they have probably done almost 1,500 genome sequences of the patients who are SARS-CoV-2 positive. And that's when we realized that even the gold standard benchmark tools do not perform with the 100% which you would need in order to actually qualify other kits to be a positive or a negative approved kits and so on. And we had to do all these things. We had to run qPCRs with multiple kits, understand what was the variability across kits in order to improve the assay. But more importantly, to improve the assay ourselves, what are the points which require more, more robustness, how to make it suitable from an end user perspective. It's always good to have things working in the lab, but when you're thinking about a kit, you need to make it work at the hands of the user reproducibly every single time. We needed a good partner. Tata Sons came up. We licensed the technology to them, and they coming from a completely different area. We needed to, handhold them for days and days, and almost months actually, to make sure that the technology is transferred. I think things look very good. So yeah, I mean, we are already, at the moment developing something which I'm really looking forward to personally, is to have something which can be done almost as a point of care or almost like a home test, which is my next, let's say, the advancement that I'm thinking of in terms of extending Paluda further.
Thanks, Dave. Mashimo-san, to you, what's your message to the researchers, how to keep all the scientists or all the students motivated during this challenging situation?
Yeah, keep challenging that. Actually, in my opinion, the scientific technology helps such situation, difficult situation. So as the former speaker said, in our case, CRISPR-Cas technology it's really like point of care testing. So we can check every day, everywhere, and even at home. So keep challenging and keep studying using anything. So good luck.
Thanks, Mishima-san. Professor Khanna, from your perspective, what's your message to the researchers today?
Look, I think, from my point of view, we were actually a little bit lucky, you can say, that we didn't suffer that much in terms of, COVID breakout here in Queensland. We've been quite sort of, we hadn't had much infection coming in. The community transmission has been almost negligible. There have been few occasional cases here and there. Most of them are actually people who have traveled from overseas. So our lab actually sort of partly shut down, but we actually continue to work in the way that what we did was we actually divided our staff into sort of working in the morning shift and the afternoon shift. And that really kept the work going. One of the major challenges for us during this period was the clinical trials that we run. We were really struggling, and particularly the ones we are running in collaboration with international collaborators, particularly in Hong Kong. We have literally, the clinical trials have stopped from that, and there have been very few recruitments. So yeah, that has been quite challenging, but I think what we have done is we've taken this time to focus on developing sort of a new platform technologies and new ideas and explore new options, what could be sort of done. And COVID came an opportunity to us to really work on that, and we really pushed that program quite hard. And we have, as I said before, we have now developed a whole blood test similar to quantiferon, but we're also actually developing a COVID T-cell therapy as well for critical ill patients, those patients who cannot mount their own T-cell response. Our goal is to actually have off-the-shelf COVID T-cell therapy available for really, really sick patients who are in intensive care and need urgent intervention, immunological intervention. So that will be something quite exciting, and we're starting to develop a process for manufacturing of those T-cells. And what we are doing is we're recruiting these recovered patients who have doing well and have no detectable virus, but they have very strong T-cell immunity, and trying to then generate T-cells off the shelf from these volunteers and use it for the patients and match it to the HLA-based system. Yeah.
Thanks, Dr. Rajiv. I will go to Sydney for this last question. How you, as well as your colleagues or collaborators from A-Star and others, how do you see you guys are keeping your teams motivated during this time?
So actually on this one, I think it's for the teams, it's really the common vision and the commitment to be able to see what is the impact of what we're doing. And that was enough to drive everyone. In fact, when very few labs were working during the period when, during the circuit breaker, for example, we continued to work. We gave everyone an option, so it was never a case of we have to. It was a case of we want to. So I think we, in fact, people were everyone in the team was not satisfied with the option of coming in every other day, taking different shifts, or every other week, different team, different weeks. Everyone wants to be able to come in and accelerate the work all the time, and we decided to go on shifts. So everyone came in every day, and we just did 2 shifts, the day shift and the night shifts. And we literally were working round the clock. And we gave everyone a choice not to have to do that. Everybody wanted to do that. It was really being able to see the impact of what we're doing that was driving everyone. It was very rewarding to be able to see launch of various different products with different commercial partners, and how they are actually benefiting different people in different countries. So not just Singapore, but being able to support other countries. So I also want to highlight, I think some of the other speakers had highlighted earlier, the importance of partnership in times of peace and in times of needs. I think during peacetime, we spend a lot of time nurturing different partnerships, and those have come in really, really, handy in critical times when we are able to really activate our partnership to be able to accelerate some of this work. DXD Hub being really a public entity, we rely on our commercial partners when it comes to really commercializing and deploying some of the products that we work with together with them. And I think especially for diagnostics tests, where it is really something that you need to continuously improve. Once you launch, it is not just, it's not an event, it's not a transaction. It is actually a continuous process. So you launch a product, you get the user feedback, and then you come back and you improve, and then you do an improved version. So because of that iterative nature of diagnostics tests, having a good relationship with your partners is extremely important, to be able to continuously have that relationship so that you can continuously improve with your partners in terms of the products that you push out together with your partners to the market. It's something that I feel also had worked really well for us. Thank you.
Thanks, Sydney. This was my last question. I just want to thank Dave, you, Sydney, Mashimo-san, and Professor Khanna for exciting discussion. I can tell you 2 things from my side. My respect for researchers increased substantially during this COVID-19 pandemic. I can see people working day in, day night to bring new innovation. So I just want to thank on behalf of Merck, not only to 4 of you, but also to all the people who are working in your labs, students, postdocs, as well as your collaborators, because of whom you are able to bring such great work to our audience today. Thank you, everyone. And we are looking for our audience to join our next session on therapeutics and vaccine development related to infectious diseases. Thank you, everyone.
Thank you. Thank you, Vibhu.
Thank you so much.